Hey biologist, Mr. Fod here. Today we're going to be talking about the properties of proteins. First up are the must-knows. We will be talking about how proteins reach their final shape, known as a conformation. This ultimately is going to determine its function. We can denature these proteins. These denaturations will cause changes in the conformation, that shape, and ultimately cause a change in its function. The denaturation process can be caused by changing in heat or pH. So first off, what is a protein and what does a protein do? It's difficult to describe. We're going to be looking at a lot of different ones of these as we go on through the course, but this is like a huge part of the AP Bio and just biology in general, this whole world. So it has a lot of different roles. It doesn't really just do one or two different things. I mean, they go on probably hundreds of different roles they actually do and functions they do. To sum it up, the best way I could put it is proteins in general, quote, get stuff done. So if you ever need to do something, like for instance, transport, you need to receive something, you want to speed up a chemical reaction like an enzyme or hold something in place, there are proteins that can do all of these various things. So we were going to be coming back to these proteins time and time again, and they're going to be these giant colorful blobs, typically purple blobs. So they might be other colors as well to indicate the differences between the different proteins. But just know that they have many different roles. There's not one or two particular ones. To make a protein, it's made up of four different levels of structures. The first are made up of the amino acids referred to as a primary structure. So this is just going to be a long chain of amino acid, this big long string. Basically, think of it like a beaded necklace or something. There are 20 different amino acids I'll showcase in just a moment. Each one of those have different properties, which are going to help determine the later structures of how the protein is then created. Each of these amino acids are held together by a specific type of covalent bond referred to as a peptide bond. And ultimately, it's going to be the holding the glue portion of the amino acid to the other amino acids. When we look at individual amino acids, there are going to be some consistencies between each of the 20 that we're going to be observing. So we can see at the diagram to the right, first off, there's something a little bit new that I want to introduce to you. It's referred to as a side chain or sometimes R group. So in organic chemistry world, on occasion, I don't want to say they get lazy, but they definitely try to speed things up. And so instead of saying, oh, and then something else, they just put an R. Or sometimes when they need to show multiple R's and they want to be able to tell, well, they're different R's, they're different something else's, they'll use an R with like an apostrophe or prime. So you might see like R prime, for instance. But this R just represents a side chain or and then something else. But the rest of the pieces of this amino acid, the amino group, that's the nitrogen with the two hydrogens, the carboxyl group, the carbon, COOH, with the two oxygens and one hydrogen, and as well as the hydrogen in the downward position from the alpha carbon, that's the one in the center, those are all going to remain consistent no matter what amino acid you happen to be talking about. The only thing that's going to change from amino acid to amino acid is that R group, that side chain, and then the something else. As we're going to look at in just a second, I'll bring up the amino acid chart. We're going to observe that they have many different properties. They could be hydrophilic, hydrophobic. They can have ionic, meaning positive and negative charges, which could lead to them being more acidic or more basic. Here are the 20 most common 20 amino acids, and please do not memorize this chart. It's just one of those things that if AP Bio wants to ask you a question about, they will give you all the necessary information there is to answer questions. The more important thing is to be able to observe these molecules and answer questions about them, not did you memorize the structural formula of tyrosine, for instance. But there are 20 naturally occurring amino acids that we can that are part of our amino acid sequence to make up of our proteins here as humans. But as noted at the top here, there are over 500 different amino acids that exist. However, we have encoded only 20 that make up our biological natures. Again, us as humans we're going to focus on. But there are vast arrays. And again, as mentioned in the previous slide, they can be hydrophilic, hydrophobic, acidic, basic, or even some that are like sulfur containing, like cysteine and methionine down at the bottom. Moving on, if we were to create our peptide bonds, we need to once again do some dehydration synthesis. we're going to dehydrate, meaning removing of water. We need to remove the OH from the carboxyl group and the H from the amino group, which are going to end up resulting in the H2O, the removal of water. Now we let the reaction carry to completion, and now we have successfully combined the the amino acids to form a peptide bond. So then we can continue on this way and to ultimately creating our polypeptide, which are many different peptide linkages. Now, once we move into multiple of these, we move into what's called the secondary level. So now we have strings of these amino acids. Those properties of the various amino acids are going to start to cause the protein to turn and to bend and to twist, and they're going to start to twist and turn and form these different shapes. Part of what's going to help determine the shape are the properties that the amino acids have. There are two major distinctions within the secondary level of the protein structure, which is the alpha helices and the beta pleated sheets. These are going to be held together, noticed by the dashed lines or hydrogen bonding. So again, depending on what types of properties the amino acids have, it's going to twist and turn and start to work itself into that conformation, the final shape that the protein is ultimately going to result in. And they can form spirally staircase looking things, the alpha helices, or kind of back and forth, these big, long, flat sheets known as beta pleated sheets. So basic principles about how the protein knows what to do. Again, it's all about the properties, which is why we focus so much on those when talking about water's relation to things, hydrophilic, hydrophobic, hydrogen bonding, all of that stuff definitely plays a part into how these proteins are going to be shaped. And as we're going to see time and time again, it's that shape or conformation that's going to determine the functionality of that protein, about whether it's functioning correctly, it's going to have a different function, or it's going to stop functioning altogether. The third level is going to be called tertiary. Now, when we start to get through this, we're going to have additional bonding of our side chains, those R groups, which are going to move beyond just the simple alpha helices or beta pleated sheets. Now they're going to start to cause more bends and twists and turns. So we can see additional hydrogen bonding, ionic, disulfide bridges, or van der Waals interactions in which are going to physically push our molecules into specific orientations or locations. Ultimately, we are going to get to what's called quaternary structures, and so this is where we have multiple regions of our polypeptides are going to be bonded together. So you can see in this molecule that we have on the right is hemoglobin. It's made up of 4 quadrants or quaternary here. And then they all come together to form the massive structure, the whole protein itself, called hemoglobin. So moving from amino acids all the way up to the protein itself, we are forming these polypeptides. Poly meaning many, peptide referring to the linkages between each of the amino acids. So again, those are covalent bonds. Again, primary structure, those are just the individual amino acids. Then we move into secondary structures, alpha helices and beta pleated sheets. Tertiary, now they start to begin folding even further. And quaternary, we are going to ultimately create the final shape or functionality where it's going to be determined in that protein. Here's a few examples and a few things I want to point out. So enzymes, you may have heard or recalled these. These are going to be things that are going to speed up chemical reactions. Like for instance, lactase will speed up the digestion of lactose, which is a naturally occurring sugar in a lot of dairy products. For intensive purposes, when talking about enzymes, they are always going to end in an ACE, A-S-E. So definitely note that in your notes that the ASE ending, like lactase, is going to be an enzyme. That's only specific for enzymes. All the other ones, for the most part here, are going to end in ins. So it's going to be something, something, something in. Like for instance, for storage function, the milk protein casein, or the transport oxygen molecules, hemoglobin, or regulating your glucose levels in your blood, insulin. So we have these in proteins. They're going to be part of the name, that ending, which is generic for proteins. Here's a few overview of some functions of proteins. Again, we'll get more into specifics as we go throughout the course, but we can have enzymatic proteins, defensive proteins, storage proteins, or transport proteins to help us. Additional hormone proteins, receptors, contractile or motor proteins to cause movement, and structural proteins to hold things together. So it's difficult to pinpoint one or two specific examples or functionalities. These things do a lot of things. So we'll get more into examples as we go along. The creation of these ultimate structures, and which is going to determine the function of these proteins, they need to be assembled properly. These don't naturally just assemble, not typically anyway. They sometimes need what's called a chaperonin, much like a chaperone, to watch and to check on its behavior to make sure it's doing everything it's supposed to be doing. This chaperonin, notice how it's an in, so it's another protein, that allows the polypeptide to come in to the chaperonin protein and allows it to fold in the proper structure. And so once you do this, now the protein has been activated once it's in the correct orientation or conformation. Additionally, what we can do to affect the conformation or shape of these proteins is that we can denature or renature. So using the chaperonin would be the renaturation process in which we are folding this into a normal functioning protein. But if we were to denature it, we could do this by altering the pH, either getting too high pH, too low pH, or the same thing with temperature. It could get too hot, it could get too cold. If the conditions are not optimal, it could cause a denaturization process. This is actually what we do while we cook our food. So this is part of the reason why it's a little easier on our stomachs to digest things, in part because we cook our food, which means we're not expending as much energy. Therefore, we get more energy out of the meal because we're not spending all that time denaturing it, using various enzymes and proteins to break everything down. So when we cook things, increasing the temperature, like for instance, like a burger or something where you have to grill it and you really got to work that heat, it's going to cause a denaturization to the protein, which is also part of the reason why you see the discoloration. It's causing a chemical reaction additionally. The biggest takeaway for proteins, I cannot say this enough, and you're going to hear me say this 1,000 times. The structure of the protein determines the function. If you change the structure, you change the function, which means it will no longer do whatever the function it is that it's supposed to be doing. So for instance, here's an example that's pretty common, and we've actually identified it to the specific example or the specific amino acid that has been mutated to cause this disorder. This is referred to as sickle cell anemia. So there's a defect in the hemoglobin protein in that the body is producing for our red blood cells. If we look at position number 6, notice how in the normal hemoglobin on top, we have GLU, we have glutamic acid, but it gets switched to VAL, which is valine. Those two amino acids have different properties. They do not have the same ones. So for instance, GLU, glutamic acid, is acidic, whereas valine is not acidic. So if we look at that, the properties are going to change, a conformation change, a change in the structure. So notice how when it works from primary to secondary, it doesn't fold properly it's not looking the same. Then it moves into the quaternary structure all the way up. Then it definitely doesn't look the same, which ultimately results in a structural change and functional change, meaning that that hemoglobin molecule is not going to carry the oxygen as it would normally. Thank you all for watching. Please let me know if you have any more questions.